Hello, my name is Eva Colleen, and I'm in the medicinal chemistry course at Cleveland University, and I'm going to dive into the new and exciting technology of CRISPR-based gene therapy. Before I get started, I want to quickly go over everything I'll be explaining in this presentation. First, I'll explain what CRISPR is, where it was derived from, and how it was developed into a groundbreaking technology which holds the ability to change the future of medicine. Then I'll go over the mechanism by which CRISPR works, how it edits genes step by step on the chemical level. Next, I'll cover real-life examples where CRISPR has already begun changing lives and pathways that are currently in trials. This will inevitably lead us into discussing the ethics, the advantages and disadvantages of CRISPR, and what the future might look like as CRISPR develops. In order to express just how valuable and powerful CRISPR technology can be, I want to preface the science with a look at who this technology holds the potential of helping. As we will dive into on the next few slides, CRISPR excels in finding and fixing single gene mutations. This means that diseases caused by a single gene gone wrong may have found their new cure. These include diseases like sickle cell anemia, a debilitating blood disease affecting over 8 million people worldwide, beta thalassemia, which affects 60,000 newborns a year, and it has even shown to make fighting cancer more accessible, which could lead to revolutionary breakthrough. This is only a few of the many diseases CRISPR could provide therapy for. So what is CRISPR? CRISPR is a mechanism used by prokaryotes to defend themselves against viruses called bacteriophages. It is a part of their adaptive immunity that works by storing DNA of the bacteriophage in order to recognize them later and attack them. As you can see in the image in the bottom right corner, the bacteriophages attack the bacteria by latching onto the cell's surface and injecting its DNA. The bacteria then becomes a host for the phages, as its DNA instructs it to build new phages. CRISPR is an acronym which stands for Clustered Regularly Interspaced Short Palindromic Repeats, which describes the section of the bacterial DNA which houses the memory of the bacteriophage DNA. As you can see in the image above, the palindromic repeats are identical segments of the DNA that can read the same forward and backward, and they are repeated multiple times. The spacers are the molecular memories of the phage DNA. Each spacer is unique and can be passed down through generations of bacteria. When we dive into the mechanism of CRISPR, the process of how CRISPR works will be further explored. But for now, I will explain how CRISPR provides immunity for bacteria. The CRISPR segment in the DNA is transcribed to RNA, which is chopped into small functional units containing tracer RNA and a spacer, which together find and bind to the attacking virus. This functional unit of RNA, in conjunction with the CRISPR-associated or Cas protein, acts as a scout in the bacteria, searching for the matching DNA to its spacer. And once it finds its correct match, it binds to the DNA, and the Cas protein snips the phage DNA into pieces, killing the virus. Now, for a while, we only knew that there was a weird sequence of DNA in prokaryotes, but had no idea what it could mean scientifically. The mysterious CRISPR DNA sequence was discovered in 1987 in the E. coli genome. As more research was gathered, it was found that very similar DNA segments were found in many prokaryotes, raising questions about their biological function. In 2007, the function of the CRISPR segments was discovered by Philip Horvath's group. And by 2011, Van der Rohe's group had reconstituted CRISPR-Cas systems from E. coli and discovered the way that CRISPR RNA was able to guide Cas proteins to specific DNA, which opened the door for major genetic engineering. Jennifer Doudna and Emmanuel Charpentier published a paper in 2012 which displayed the programmable Cas9. This directly enabled human genome editing with CRISPR. Now, let's take a deep dive into what mechanisms allow CRISPR to work. First, we must understand the chemical components of CRISPR. These include Cas proteins and guide RNA. The Cas9 proteins are composed of 1,368 amino acids and work as the genetic scissors to cleave the target DNA. Guide RNA is made up of CRISPR RNA and transactivating RNA, or tracer RNA. The CRISPR RNA is made up of 18 to 20 base pairs, which pairs with the target sequence. This is the key to modification that is used during drug production, since it can be changed to pair with any set of base pairs. Tracer RNA is a series of loops that acts as a binding scaffold for the Cas9 nuclease. The mechanism begins with our guide RNA binding to Cas9. This occurs through ionic bond formation between positively charged arginine and lysine residues on the Cas9 protein and the negatively charged phosphate backbone in the guide RNA. The complex is then stabilized through hydrogen bonding between amino acid side chains and the guide RNA bases. These reactions will reveal two catalytic sites within Cas9. These are HNH and RUVC, which will serve as our genetic scissors later on. PAM, a specific DNA sequence following the DNA target, is recognized by arginine 1333 and arginine 1335 through hydrogen bonding with guanine 06 and N7. This allows the breakdown of the double helix through pi bond stacking disruption, ultimately providing a site for strand invasion by the guide RNA. The guide RNA bonds to the target DNA through Watson-Crick hydrogen bonding, displacing the non-target strand into the active site of RUVC. Now that the complex is in place, catalytic cleavage can take place on the target DNA. The HNH endonuclease within Cas9 attacks the target strand first by activating a water molecule as a nucleophile to attack the phosphodiester bond of the DNA. RUVC, on the other hand, attacks the non-target strand of the DNA by another hydrolysis reaction, resulting in a staggered double bond strand break. Both of these reactions are dependent on the magnesium as a metal ion catalysis. Finally, cellular repair occurs by either non-homologous end joining or homologous directed repair pathways. The first is more common and is faster, but the second is more precise, but requires a DNA template, limiting when in the cell cycle it can occur. Because of this reparation of the products, the reaction is irreversible. Using CRISPR in drugs is revolutionizing medical possibilities. Sickle cell anemia is a blood disorder affecting 8 million people worldwide. It is a genetic disorder caused by a mutation in the gene coding for beta globin. This causes the molecules to stick together and distort their shape, from their disc shaped to the crescent shaped. This disease causes major repercussions on the body. Sickle cell patients have extremely painful episodes due to blocked blood flow. They also suffer from anemia due to the faster breakdown of red blood cells. This can bring on a whole new onset of symptoms, including fatigue, increased risk of infection, and organ damage. Luckily, these patients are looking at CRISPR as a new treatment. The first patient to receive this treatment was Victoria Gray, and it demonstrated a successful treatment. Her own hematopoietic stem cells were removed from her bone marrow, then edited with CRISPR and injected back into her bloodstream. The CRISPR drug reactivated her fetal hemoglobin genes, which are usually only produced during fetal development. This allowed her blood cells to return to their natural shape and put an end to her suffering. There are many other amazing examples of what CRISPR gene editing has already begun. One situation that was highly controversial was the first CRISPR edited embryos. He Jiankui edited the CCR5 gene to make them HIV resistant since their father was HIV positive. The issue with this was that it was an extremely risky procedure as it could have affected other parts of their genome, and there were already alternative ways of HIV prevention. This opened a world of questions about the ethicality of CRISPR. People ask if CRISPR should be used in germline cells or not, because this would edit an entire generation. They also question what we would do with this power and whether it should be done or not, acting as God, and could even play into eugenics. Looking forward, this could potentially provide a cure to cancers and even prevent genetic predispositions to things like obesity and addiction. This technology is absolutely revolutionary and could cure and prevent tons of diseases or unfavorable outcomes in health. We must be careful as a population with what we choose to do with it.